Laboratory Investigation is releasing 2 special issues on vascular biology, featuring exciting new research as well as overviews of the field. Many of the papers focus on cardiovascular disease, which is the leading cause of death in Western society, causing 17.3 million deaths. Cardiovascular disease is usually associated with a buildup of fatty deposits in the arteries called plaques, a condition known as atherosclerosis. The narrowing of arteries that supplies the blood to the heart can lead to the damage of heart muscle, peripheral artery disease, and ultimately stroke, heart attack, and heart failure. Risk factors for cardiovascular disease include a high-fat diet, high cholesterol, and high blood pressure, and eating foods high in fats has been shown to trigger heart attacks in patients with atherosclerosis. In a paper in the first special issue, Benson et al. Demonstrate that a single high-fat meal in humans can cause pathological red blood cell remodeling and elevates levels of the compound MPO, which may be able to enter the blood vessel wall, trigger oxidative stress, and destabilize plaques. These findings may have implications for the short-term impacts of high-fat meal consumption, especially in patients with atherosclerosis. Vascular smooth muscle cells regulate the tone of arteries and provide structural stability required for oxygen delivery to tissues. However, they can also affect plaque formation by switching from a quiescent state to a migratory proliferative state in which they create thickened vascular walls and help form fibrous plaque caps. In a review in the first special issue, Durgan and Straub look at the evidence that reactive oxygen species can facilitate this switch and outline mechanisms by which it may occur, including Knox pathway signaling, the fission or fusion of mitochondria, and the SGC, CGMP, PKG pathway. In a paper in the second issue, Davis-Knowlton et al. Studied smooth muscle cells found in human atherosclerotic plaques that had been removed during surgeries. They found that SMCs from lesions had distinct proteomic profiles and altered responses to a signaling system called the Notch pathways. The subset of proteins they identified could work as functional markers of diseased SMCs. Also in the second issue, Dotla et al. Investigate the role of a protein, POLDIP2, in SMC proliferation. In mice, they demonstrate that wire injury of the femoral artery leads to proliferation of SMCs and a subsequent thickening of the blood vessel lining, but that this is impaired in mice with POLDIP2 knockdown. They also show in vitro that downregulation of POLDIP2 leads to reduced serum-induced proliferation. Disruption of POLDIP2 binding could therefore be a novel therapeutic strategy to control unwanted proliferation. Gut microbes also play an important role in cardiovascular disease. TMAO, which is metabolized by gut microbiota from dietary choline, has been shown to increase vascular inflammation and induce atherosclerosis. In another paper in the second issue, Lee et al. Investigate the role of TMAO in cardiac hypertrophy, a common stage of many cardiovascular diseases where the heart muscles increase in size due to cardiac overload and which can eventually lead to heart failure. The authors found that TMAO directly induced cardiac hypertrophy in vitro and in vivo, and that treatment with antibiotics inhibited this effect. They suggest that a TMAO model of cardiac hypertrophy would be simple, stable, and low cost, and that targeting TMAO generation could be a potential therapeutic approach. In patients with coronary heart disease, cardiac function often declines until heart failure, a terminal outcome known as Ischemic Cardiomyopathy. But 40% of the heart muscle affected by narrowed arteries remains viable by reducing protein expression and decreasing energetic demand. The cardiac tissue also reverts to a fetal-like state, and cells metabolize glucose over fatty acids. Epigenetics could be one mechanism for this change. In the second issue, Pepin et al. Found that this metabolic shift was linked to DNA methylation in patients with ischemic cardiomyopathy. This is where methyl groups are added to the DNA molecule, which can alter transcription. They found that an epigenetic regulator called EZH2 could be involved in this process and suggest it could be a useful target for further study. The body's response to narrowed arteries and inadequate blood supply to organs, known as ischemia, is to generate new blood vessels to bypass the obstruction. One way of doing this is arteriogenesis, the remodeling and enlargement of small arterioles into large arteries. This process depends on macrophages, which infiltrate the area and release molecules that can stimulate arteriogenesis. Osteopontin is one such molecule that mediates the creation of new blood vessels after ischemia. In a paper in the second issue, Lee et al. Look at 3 different isoforms of OPN expressed in humans and found that they have different effects on arteriogenesis in vitro and in vivo, in part by affecting macrophage accumulation, migration, and survival. Human OPN isoforms may represent novel therapeutic targets to improve neovascularization.